Hi, I'm Catherine, and I'm from the main lab at Washington State University. I'm the developer of MapViewer for the Genome database for Rosacea website. Thank you all for the opportunity to talk about MapViewer today. In this presentation, I would like to show you how to use MapViewer to view and compare genetic maps in the GDR website, introduce you to several of its features, and show you upcoming functionality. Genetic maps are integrated into the Genome Rosacea database with publication, contact, and associated trait information from the data provided by authors in peer-reviewed papers and curated in GDR with standardized marker, primer, and QTL names. MapViewer is accessible from several different locations in the GDR website. There are several integration points for MapViewer, and it can be accessed from the Tools menu in the header and Map Overview page. In the Map Overview page, the MapViewer shows a summary graphic of all linkage groups. By clicking on a linkage group, it opens a more detailed view. The MapViewer displays linkage groups from different maps and organisms. Complete linkage groups are displayed on the left, and one can dynamically select and magnify specific regions on the right. The magnified region can be adjusted by dragging and resizing a window on the linkage group on the left. The linkage groups can be compared, showing correspondences between common marker features. And the color legend for the marker types is indicated below the linkage group. Each of the features hyperlinks to an individual marker page. To view more information about each marker, hover over the marker name with the pointer to see a tooltip summary, or click on the marker to open the marker overview page. Also, clicking on a QTL will take you to the QTL overview details page. The Marker Overview Details page gives the marker information, and in the Map Positions tab, the MapViewer table column links back to the linkage group that contains each marker in the MapViewer. Below the linkage groups on the MapViewer page, the control panel is used to configure which linkage groups are displayed, as well as their viewing options. We can select different reference and comparison maps, configure options for marker and QTL display patterns and visibility, and change color display of different marker types. The ruler and marker positions can be toggled on or off. In the on position, each marker displays its own marker position label. The Expand QTL option allows QTL labels to directly follow their QTL lane, rather than in a single vertical column following all QTL lanes. After updating these settings, you can press the Submit button to display the changes. Here we can see marker correspondences between linkage groups from different maps. Zooming in on the region where the correspondences exist on the left, linkage group, or the right comparison linkage group enables to focus on specific relationships of markers in common between the two linkage groups. In this video, we can see how the zooming on each side exposes further details for each linkage group along with correspondences. The MapViewer in GDR can be used to access 316 maps containing about 5.5 million markers and roughly 2,800 QTLs for 106 traits, with more data being added from publications on a regular basis. The correspondence matrix and DOTPLOT features are available and provide a more comprehensive representation of where correspondences exist across multiple maps and organisms. The dot plot shows the relative positions of common markers, and clicking on a map point will open the corresponding page for that marker. The correspondence matrix displays the number of marker correspondences for linkage groups between two maps, or between a single map and all maps in a species. Selecting a grid element will click through to the map viewer displaying that particular map comparison combination. Upcoming functionality will add a genome view to display markers in common between genetic maps and genomes. The genetic markers are displayed on the left side of the genome view, and the number of genes and mRNA are shown by the size of the blue bar on the right. Clicking on a gene or genetic marker will take you to JBrowse, acting as a bridge to allow further viewing of genes and other features in JBrowse. Thank you.